Bismillahirrahmanirrahim. Assalamu alaikum everyone. I welcome you all on behalf of Pakistan Endocrine Society in today's session. My name is Dr. Sumera Batul and I'm an assistant professor and consultant endocrinologist at Al Khan University Hospital, and I will be your moderator today. So let's start the session with the recitation of Holy Quran.
بسم الله الرحمن الرحيم قل هو الله أحد الله الصمد لم يلد ولم يولد ولم يكن له كفوا أحد.
Jazakallah. So today's session is a second webinar of our series Demystifying Pubertal Disorder. And today's topic is normal puberty and a brief overview of embryology, genetics of the reproductive system. As you all may recall, our first webinar was on 27th July and it covered male and female reproductive physiology. So today's program will build upon that foundation. We have our worthy panelists with us today. So let me introduce you with them. First, I am honored to introduce our distinguished panelists. We have Dr. Aisha Sheikh with us. Dr. Aisha Sheikh is the president of Pakistan Endocrine Society and consultant endocrinologist at Al Khan University Hospital. She is also a recognized tutor at University of South Wales, UK. Dr. Aisha Sheikh has significant expertise in the field of endocrinology, particularly in the gestational diabetes and women health. Our second panelist is Dr. Khadija Humayun. Dr. Khadija Nuzhat Humayun is a professor and consultant pediatric endocrinologist in the Department of Peds and Child Health at Al Khan University Hospital. Dr. Khadija initiated the first structured fellowship program in pediatric endocrinology in Pakistan and has played a key role in its accreditation from CPSP. She has multiple awards for her contribution to medical education, including the Best Teacher Award and Innovation in Teaching Award from AKU. Our third panelist is Dr. Sadia Salman. She is head of endocrinology department at Allama Iqbal Medical College and Jinnah Hospital, Lahore. She is the General Secretary of Pakistan Endocrine Society and a member of American Association of Clinical Endocrinologists. Dr. Sadia has extensive experience in endocrinology and has published more than 20 research papers in national and international journals. Our fourth member of the panelist is Dr. Mohsina Noor Ibrahim. She's a professor of Peds endocrinology with 20 years experience in one of the largest hospitals of Karachi, NICH. She is a CPSP supervisor for general Peds and Peds endocrinology and has more than 50 research papers in her credit. She is also the Trier Secretary of Society of Peds Endocrinology and Diabetes of Pakistan. I welcome all the panelists in today's program and thank you all for taking out time for today's session. Moving on to our speaker, we have a very renowned and a dynamic personality with us, Dr. Salman Kirmani. Dr. Salman is an associate professor and a consultant pediatric endocrinologist at Al Khan University Hospital. He has a special interest in pediatric genetics and metabolism. He's a diplomat of American Boards of Medical Genetics and diplomat American Boards of Peds Endocrinology. He has a background as a past education chair at Mayo Clinic and is a director of the AKU Center of Excellence for Women and Child Health. Dr. Salman Kirmani is a well-versed in pediatric endocrine and genetic issues, and he will present on normal puberty and a brief overview of embryology and genetics of the reproductive system. Over to you, Dr. Salman, and thank you for joining us today.
Assalamu alaikum everyone, and thank you so much for that very, very kind introduction. And thank you to Dr. Aisha and the rest of, Dr. Sumera and the rest of the Pakistan Endocrine Society for inviting not only myself, but I think the entire SPED Pakistan team to do some of these talks. And we're very excited. I think this is a great cooperation between two societies that have so far worked independently, but I think this is our first joint venture together. And I think this will be very, very beneficial for not only the adult endocrine community, but also the pediatric endocrine community and all the trainees that will come out of it. So I have been asked to talk about a tough topic overall, but my goal is to make it as simple and as easy for everyone, because that's how I understand these things. I'm hoping that my screen is being shared. If not, please let me know if I have to do a share screen, or is it already on? Can people see my screen?
So you have to share your screen.
Okay, so let me see. Let me put the share on again. Okay. All right. Can people see my screen now?
It's visible.
Yes, yes, we
Oh,
can see now.
perfect, perfect. Sounds good. So I'll begin, and I'm not exactly sure how many people are there in the audience, but if someone can let me know about comments in the chat section, because I've just minimized my mini screen, all I can see is the slides. And if there's something that needs to be addressed while I'm talking or needs to be clarified, I'm very happy to be interrupted. I don't mind. So I'll let the moderators do that in case that's needed. Otherwise, we'll do questions at the end, and we'll keep it short. It won't be a very long talk. So we're going to talk about the embryology and genetics of puberty and the reproductive system, knowing that most of us are practicing clinicians, and our perspective really comes from the fact that we need to know this so that we can use that information clinically. There are lots of facts and lots of concepts that are there, and I will concentrate on the bare essentials which need to be remembered or referred back to, because you can't remember everything. But as long as the principles are known, people can then conclude, and a lot of the interpretation of our genetic and hormonal test results depends on our understanding of physiology. So it's important to kind of understand that and take it forward. So we'll begin, and so I have no disclosures. And really, two basic things that I'm going to go over. One is a very, very interesting question, which is what turns puberty on? Because that has, really not been completely answered yet, but lots of insights, especially over the last 10 to 15 years, have come in, and a lot of the disorders of puberty now are being clarified based on that information that is out there. And then I'm going to switch gears a bit and talk a little bit about the genetic and hormonal control of the development of the internal and external genitalia, which obviously play a huge role in the disorders of sex development. Today's talk is not about diseases at all. It's all about physiology and embryology. So I'm guessing that at a later stage, we will have chances to concentrate on that. And I know that a lot of people will have questions regarding that, and we're happy to answer that. But just to let you know, that none of the material today will actually deal with diseases. It will deal with concepts in physiology and embryology. So this is an important slide, because it goes to show that there is not one thing that influences the timing of puberty. Your background genetics clearly play a role. Ethnicity, we know lots of data out there that ethnicity plays a role. We know that people of Hispanic origin, people of African origin, South Asian origin tend to go into puberty earlier compared to people of European descent, and so forth. And there's actually very interesting reasons for that if you think about it from an evolutionary standpoint, right? Because people have asked this question that, why are South Asians going into puberty at age 9 or 10, whereas, the Western counterparts are going into puberty at 12 to 14? And the answer lies in the fact that we know that life began in Africa, and then there was northward migration. And as the migration happened, you were going towards more, colder climates, etc., and so forth. In the African continent, especially in the equatorial regions, food was aplenty. It was very easy to hunt and to gather and to feed communities. And so when those initial communities were evolving, it made sense that they needed to procreate and increase the human race. And so the earlier the puberty happened, the more reproductive potential a woman had for her lifetime. And so you could, create more humans and so forth. And that's what happened. And then, migrations happened because man became adventurous and there were other pressures leading people to go northwards. And obviously, there were always people who were evolutionarily, selected to start puberty a little bit late. Genetically, there were differences and variations. And evolution basically selected for those individuals who moved north, because the people who moved north had less resources, they had less food. Winters were much longer and much harsher. So it didn't make sense to spend your energies getting pregnant and going through puberty. It made more sense to save your calories to make your heart beat and your brain function. So people who survived more in those harsh environments were the ones who went into puberty late and actually had less reproductive potential early in life. So that's why there is a segregation today, where people of Caucasian descent who are descendants of people from Northern Europe are the ones who are actually going into puberty slightly later, whereas people of African, Middle Eastern, Hispanic descent who are equatorial are the ones who have retained their ability to go into puberty early. So very, very interesting concept right there. Then, you have prenatal conditions which influence the way things go. And that's very clear. We know that the fetus is under many, many influences. And we'll talk a little bit about those which come under epigenetic mechanisms. The nutritional status of not only the mother, which affects the fetus, as well as the individual going through puberty will clearly affect. We know, for example, that in the most extreme cases, for example, anorexia nervosa, you actually do not go through puberty at the right time, and you actually end up with primary amenorrhea. And that's for the same reason, that you need a baseline nutrition to get all the things lined up for puberty to happen at a normal pace. Maternal conditions, obviously, if the mother has any illnesses of any sort, any other things going on, that will affect how the fetus is programmed to go into puberty later on. And then many, many environmental factors. Nutrition could be considered an environmental factor, but also climate, temperature, etc. And then there is a whole host of what we call EDCs, endocrine disruptor chemicals. And they are chemicals that are present in the environment. Some of them are pesticides and some other synthetic chemicals that are present ubiquitously everywhere. And people feel that any recent trend in the timing of puberty or changes, etc., may actually be related to those endocrine disruptors rather than any genetic factor, because genetic factors take a long time to change. Finally, epigenetic mechanisms. And I really want people to understand the difference between genetic mechanisms and epigenetic mechanisms. So by definition, epigenetic means above genetics. It is the layer above genetics, the layer that controls what genes get turned on and what genes get turned off. So even though genetically there may be two people who are identical in their pubertal timing, but it may very well be that if certain key genes are turned on or turned off, the people with two identical types of genetic predispositions may go into puberty at different times, because the layer above genetics, the environmental factors, have influenced which genes get turned on and which genes get turned off. And all the above things, ethnicity, but prenatal conditions, nutritional status, maternal conditions, environmental factors, all in the end are factors that epigenetically will determine what happens to puberty. So I hope that was clear. I know that's a lot to talk about for just one slide, but I felt it was really important to give you a really wholesome view of all the different things. And now you can appreciate and imagine why we don't have a clear answer for what turns puberty on, because there are so many different nuances and so many different things to sort out. Very, very hard to put your finger on it, and probably will remain so, because the factors are ever changing and we're still gaining our knowledge in this area. Then, we'll come back to our sort of understanding of a simple physiology again, which means that, when you're talking about the pubertal axis, you're talking about three main layers of endocrine glands. The first is the hypothalamus, second is the pituitary, and the third are the gonads, which in the female is the ovary, and the male are the testes. As you can appreciate, within the hypothalamus, there are neurotransmitters that are controlling the central axis, which is the GnRH axis, the gonadotropin releasing hormone axis. And you can see that there are certain neurotransmitters that will have a positive influence on the secretory potential of GnRH, whereas others will have an inhibitory effect. And this combination of these different pathways, which is the KNDY pathway, the kisspeptin pathway, the glutamate pathway, or the GABA pathway, ultimately, are the main influences through which GnRH starts its influence on the pituitary. And this is sort of the orchestra that does happen once the pubertal axis becomes active in its own right. At the level of the pituitary, once you have gonadotropin releasing hormone, it will influence the pituitary to release both LH and FSH, which then go and influence the gonads, as is mentioned over here. Most of you know this already. And then the gonads will make their respective hormones, estrogen, or in the female, testosterone in the males, along with obviously both the gonads making inhibin B, and they have a negative influence back on the pituitary, as well as a influence back on the hypothalamus. So you can see how this feedback loop goes. So I just wanted to go over this physiologically, but then have everyone in the audience appreciate the neurotransmitter influences. And it's, again, not one particular influence, but many influences, some positive, some negative, that then control this in their own right. I think it's also important from a physiological standpoint to understand that what happens in pre and postnatal life. You can see both the graphs have a 0 to 9 months gestational period. Then there's birth, and then, you start the postnatal life. And as you can see, there is a concert of hormonal activity going on prenatally. And then, the phrase that everyone is born in puberty is indeed correct, because you can see how LH, FSH, testosterone, estrogen are elevated right at the time of birth, and then, settle down over the first year or so of life, with some up and down initially, but then they remain quiet until puberty decides to, wake up again. And then you see a more constant level of hormonal influence in the male, whereas the cyclical hormonal changes in the female, which lead to the menstrual cycle happening as it does, and then a very clear menopause in the female, with lowering of the estrogen levels and AMH levels, and rise of the LH, FSH, whereas in the male, there is a more steady decline slowly after the age of 50 in the levels of testosterone and inhibin B, and the rising LH and FSH tell you that the axis is finally going down. So again, important to understand that embryologically and physiologically, to appreciate when you review test results, etc., and you now realize why there's such a variation in the timing of the hormones, especially in infancy, where you might catch mini puberty and see some of these hormonal influences right there. Then I think it's really important to understand from a genetic standpoint what modulates this GnRH pulse, because that's a very important concept, right? It's not a constant influence of GnRH that releases LH and FSH. It has to be pulsatile. And what controls that pulse is this number of genes and the downstream products, the protein products of these genes that allows that pulsatility to happen, because it is that pulsatility that is truly physiological and leads to appropriate hormonal influences at the level of the gonads. Here again, you can appreciate there are different networks and different systems in place, and the genes that are there, whether it's kispeptin or the KIS1 gene or the KIS1 receptor gene, or in fact, the genes for the neurotransmitters, glutamate, leptin, GABA, etc. In the end, they're the ones that will influence this pulsatility, and any variation in these genes will cause a normal physiological variation in the pulsatility. If you have a mutation in any of these genes, obviously, then that will be an interruption to the normal process and may, cause either precocious puberty or hypogonadism. So as an example, the MKRN3 gene is a very important gene in central precocious puberty, and any mutations in that gene are the commonest cause of central precocious puberty. So I mentioned that because some of you may read papers or may be familiar with that, and it's important to appreciate all that goes on to make sure that pulsatility at this axis is normal. I think it's also important to keep in mind how the environment will influence this neuropeptide control. So this diagram tells you very simply that you can have two extreme states. And again, I'm just using these extreme states as examples. So one is a state of energy excess, and the other is a state of energy deficiency. So if you take the first one, energy excess, you can see that that energy excess, the caloric excess, will change the epigenetic landscape and will allow certain genes to exert more influence and certain genes to exert less influence. So, for example, KIS1 is upregulated when you have energy excess. That then leads to the mTOR pathway being activated, and then the downstream effect of that is more or earlier pulsatility of the GnRH neurons that then lead to early puberty or advancing puberty. So that's why you sometimes tend to see obese individuals having earlier onset. It used to be thought that the obesity was driving the puberty, or the early pubertal changes were being driven by just peripheral conversion due to excessive adipose tissue. But now turns out that the actual energy excess actually has epigenetic influences on the genes that will cause the pulsatility or early pulsatility of the GnRH neurons. The other extreme then, energy deficiency, as you can see, amplifies a very different type of pathway. And here, KIS1 is less active, and the AMPK pathway is the one that is able to, exert its inhibitory influence on the GnRH neurons. And what that will then do is it will induce a break onto the pulsatility, so that puberty is delayed until the time that calories are sufficient enough to go back again. And it makes absolute physiologic sense, as I mentioned earlier, that if you're struggling to have enough calories to keep your vital organs alive and the vital functions on, you do not have the luxury to go into puberty. You don't want to go into puberty because you don't want to become pregnant, because you don't have the energy to sustain that reproductive capacity until you at least reach a bare minimum of nutritional status. So it's very interesting that research over the last few years has really clarified a number of these nutritional influences, and a lot of hormones such as leptin from adipose tissue, etc., and so forth, are influencing how this happens, and when puberty should or should not start. So again, I hope that that gave everyone a very good example. Again, this is not a comprehensive diagram. It just is an example of two extreme situations and how they influence things. I think the other thing to now look at is what our genetic studies or genome-wide association studies have told us about the genetic influences on the pubertal timing across your lifespan. And that is very clear. So the genes in blue are the ones that have typically been associated with precocious puberty, whereas the genes in red are the ones that are typically associated with hypogonadism or delayed puberty. So clearly there are positive and negative influences. And now, since we know enough about these, our patients who present at some of these extremes, we can now test and try to find a genetic cause. And we're quite successful now because of multi-gene panel or whole exome sequencing. At the level of the hypothalamic pituitary axis, you can again see how these genes influence those functions by encoding the proteins that are either neuropeptides, actually mostly are neuropeptides, or receptors for those neuropeptides that will then cause the influence. So again, endocrinology is all about hormone and receptor, ligand and receptor, and it is very clear that any genetic influence either on the ligand or on the receptor will or has the potential to cause either earlier or later onset of puberty. And again, this is again an example of what we know today about the genes associated with central precocious puberty. Loss of function mutations in MKRN3, as I mentioned, one of the most common causes of central precocious puberty, whereas gain of function mutations in KIS1. So again, this is another concept that I want the non-genetics people in the audience to appreciate, that we think of mutation as something that causes dysfunction of the gene. And for the most part, it's a simple understanding. But one has to realize that different types of mutations influence the gene differently. Gain of function mutations actually increase the protein production capacity of that particular gene. And if that influence is negative, then it will cause an inhibitory effect on the axis. If that influence is positive, it will cause a positive influence on the axis. So it truly depends. And the same thing with loss of function. Loss of function will either, you lose the inhibitory function or you lose the stimulatory function. So I think there are nuances to mutations as well. Just simply saying there's a mutation in so-and-so gene may not truly help you understand unless you know what is the effect of that mutation on the actual protein or the hormone that it is encoding. So it's really, as you can see, DLK1, KIS1 receptor, KIS1, and MKRN3. Those are the four main genes that we know today, and we're learning much, much more. So I think because central precocious puberty is a pretty extreme example, we've learned about these genes over there. Same thing with hypogonadotropic hypogonadism, which is an extreme example. We know the genes there. What causes variation within the physiologic range is a much, much tougher thing to understand. And I think those are where more studies are needed to increase our understanding. So I'll summarize this first half of the talk by saying that genetic mechanisms causing variation of pubertal timing are quite complex and only partially understood. I'm sorry, I failed to put in the D over there. Some genes act as promoters while others act as brakes. And as I've just shown you in the last slide, some genes have gain of function mutations and some have loss of function mutations. So, if you have a loss of function mutation in a brake, you've got a double negative, so you've got a positive, so that's kind of how you have to think about it. And then finally, epigenetic mechanisms that control the genes, clearly are very, very active in fetal life, but actually influence puberty throughout infancy and childhood. And really, any time during that window, you can have issues. So with that ends the first half of my talk, and I will now switch gears to the second half, but we can then take questions on everything at the end. So now we're just going to briefly go into the embryology of the reproductive system. The way I try to explain very, very simply is that, females begin life as XX because you get one X from the sperm and one X from the egg, whereas males will begin life as an XY, where the Y will definitely be from the sperm because females will only contribute the X. So this is how it works. If you look, and I'm sorry that the slides are slightly blurred, but basically up until 5 weeks of gestation, you have what we call a bipotential gonad. Bipotential gonad means if you histologically were to take a look at the gonadal tissue, you would not be able to tell whether this individual is XX or XY because they will look identical. All you will see are genital ridges in section, and there will be no clear hormonal influence yet in the development process. We then move to the 6th week, and this is where the bifurcation starts happening, as you can see in the diagram as well, that, the genital ridges are maturing, and you have what we will call the primary sex cords within those genital ridges, and the primitive germ cells will start populating right there. Again, this is at 6 weeks of gestation, and we're still at the stage of what we would call a bipotential gonad. Again, no difference right now between XX or XY. And now I've given you a snapshot of week number 8, where there should be very clear differences. So the bifurcation has happened now. The male pathway is separate from the female. If you look at the male pathway, what you've got is you've got development of the Wolffian ducts. The gonad is differentiating into testes cords. There is a remnant of the Mullerian duct still there, which you can see, but it's mainly the testes cords that are determining this as the male gonad, and the Wolffian ducts are developing further. In the female at 8 weeks, the gonad is differentiated into the primitive sex cords, not the testis cords, but the sex cords, which are different because the testis cords will then make the internal seminiferous tubules that will then go on to connect with the vas deferens. So in the female, the primitive sex cords is what will make the ovarian tissue. You will have cortical cords in there, and then the primordial germ cells will be residing within this tissue. There will still be a remnant of the Wolffian duct, which you cannot differentiate yet from the male, but the Mullerian ducts will actually grow in the later weeks. Those will still be present. So to understand, and we'll come back to those diagrams in just a second, that, the Leydig cells in the testis tissue from 8 weeks onwards are basically making testosterone under the influence of placental HCG. So if you have testicular tissue, HCG will act on it and will make testosterone, which will then cause the stimulation and maturation of the Wolffian ducts. At the same time, the testosterone that is being made in that male gonad under the influence of 5-alpha reductase, the enzyme, will basically get converted to DHT or dihydrotestosterone, and this will form the external genitalia. And at the same time, the Sertoli cells in the testicular tissue will make anti-Mullerian hormone or AMH, which will actively cause regression of the Mullerian ducts. So you need these active hormones, testosterone, dihydrotestosterone, and anti-Mullerian hormone to have the complex male genitalia that you need both internally as well as externally. And now if you take a look at the external genitalia, this is again at 9 weeks, you can see a clear difference. The phallus is there in both sexes, but it's a much more prominent phallus in the male, again, under the influence of testosterone. There is a urethral groove, but it is very small now in the male, whereas quite large in the female, as you can see. There's still an opening there. It's supposed to be an opening. And at 9 weeks, you will still see labioscrotal folds in both sexes. Those will get differentiated a bit later. If you look histologically internally, you will now see very, very clear differences. You can see now that under the influence of the male hormone, this is now at 16 weeks. So this is where genital development internally is complete. You can see now a connection between the testes cords and the Wolffian duct. So this is where your connection between the seminiferous tubules and the vas deferens has now taken place. And the Mullerian duct is just a rudimentary structure because AMH has caused it to be regressed and just be present, like I said, in a rudimentary state. In the female, the Wolffian duct has regressed. As you can see, you don't see the connection over there with the gonad. And what you can't see in this section is that the Mullerian duct has actually developed further, and the Mullerian structures are the ones that will go on to form the uterus. The ovarian follicles have clearly developed by 16 weeks, and they have internally matured as well. So the way to look at it and to simply understand it is that initially you have an undifferentiated gonad with both Wolffian and Mullerian structures present. And once the male pathway is taken, and we'll talk about what influences that, you will differentiate that bipotential gonad into seminal vesicles, vas deferens, epididymis, and testes. Whereas in the female, those same initial undifferentiated structures, typically under very little or no hormonal influence, will develop into the uterus, the fallopian tubes, the ovary, and the vagina. So I think you can see that on a skeleton level, the structure is quite similar. But if you look at it in a much more complex fashion, there's a night and day difference between what's happened in the male and what's happened in the female. And that's, again, genes and hormones controlling this. If you look at it from an external standpoint, you can see, again, in the undifferentiated state, you have the phallic tissue, the glands, the genital tubercle is the entire mound. You have the urethral tissues, the groove, and the anal pit, all present within that area. And again, under the right influences, you will develop the male external genitalia. As I mentioned, again, DHT is clearly required for that, and the male gonads will develop internally and then descend. Whereas in the female, you can see that you will have clitoral tissue because the phallus will not grow as it does in the male because there's no male hormone influence, and there will be a urethral meatus along with a vaginal opening and both inner and outer labia. The anal tubercle and tissue will be present in both sexes, as you can see. So I think some central concepts that I want everyone to take home are that you begin again with a bipotential gonad. You have both the mesonephric and paramesonephric duct. if the Y chromosome is present, which basically contains the SRY gene or the testes determining factor gene, you will develop testes which will make androgens, and the androgens will drive male development. On the other hand, if you have no Y chromosome, so absence of the Y chromosome will cause the development of the ovary, although we know that there are positive genetic influences on the ovarian tissue. So you need a certain number of genes to make normal ovarian tissue. But at least the internal genitalia and the external genitalia of the female will develop without any, male hormone. So the SRY is actually considered the switch that diverts everything towards the male pathway, and that makes sense. The androgens and the AMH are essential for normal male development, and you need testes to have male external genitalia, because the testosterone coming from the testes will be converted to DHT, which will then, cause the male differentiation. And presence of the ovaries is not essential for female external genitalia. I think that's the clear take-home message, and that's why females with Turner syndrome, for example, even though they have, streak gonads, streak ovaries, their external genitalia will be completely normal. So you don't need normal ovarian tissue to make external genitalia female, but you do need normal testicular tissue to make normal male external genitalia. This is just a quick summary of the genes involved in male gonadal development. And the only reason I've listed these is that some of these genes are clearly associated with disorders which we all deal with as pediatric endocrinologists, whether it's, the Wilms tumor, one gene, the WT1, that causes the Denise-Trash syndrome, or a number of abnormalities in SRY, etc., or SOX9, SF1, DHH. All of these are there. The main thing, again, is the bipotential gonad turning into the testes if you are XY, ovary if you're XX, and the appropriate hormonal milieu that will then lead to male development. And on the female side, again, I know that most people end up saying that female is the default pathway, but you actually do need some active genes that come from the ovarian tissue that are also considered the anti-testis factors, meaning that they actually actively divert things towards the female pathway. And we're learning more and more about these, and some of the DSDs that are yet, under diagnosed possibly have abnormalities within these genes. So again, I will end by summarizing that all fetuses have a bipotential gonad until 6 weeks of gestation. The presence of a functional SRY gene leads to the formation of testes, which creates the hormonal milieu for male internal and external genitalia. And that the female developmental pathway, although considered by some to be default, actually requires the presence of a number of important genes, and we're learning more and more about that. And, from the pathological side, the DSDs, or the differences of sex development that we see, are really the extreme manifestations of these variations in these genes. And that the normal variations that we see, minor things like hypospadias, etc., and so forth, may have minor variations in these genes that truly we don't understand today as mutations, but maybe later we will appreciate as causing the differences we see within the physiological spectrum. So with that, I will end my talk, and I'm happy to take questions. And I'll leave the screen share in case people want me to go to a particular slide. So happy to take questions. Thank you very much.
Thank you, Dr. Salman, for such an insightful presentation on a very complex topic. You have explained the genetics part so beautifully. It's really very impressive.
Oh, thank you very much. This is one of the things I love to do. But yeah, it's something that it's a bit of familiarity. It's very hard to And I keep telling people, no need to remember the genes, no need to remember things, remember the concept and then keep coming back to it and it'll be fine.
Exactly. So we have, I would like to invite our esteemed panelists to share their comments and their thoughts about today's topic. Dr. Aisha Sheikh, can you please comment on this?
Thank you, Dr. Salman. Can you hear me?
Yes, we can hear you.
So thank you, Dr. Salman, for such a comprehensive and succinct talk on a very difficult topic. So we being adult endocrinologists, we always can consider embryology and genetics, things which actually are outside our domain. And you have made such a wonderful presentation and discussed all those complex pathways in a very simplified manner. And I'm sure our audience have benefited from this as well. And we're really looking forward to our ultimate full day symposium where all of you will be joining again as faculty members, presenters, workshop facilitators that will further enhance the clinical aspect of puberty and its disorders, because this is what these two webinars will ultimately lead to. Since without this understanding, going on to the normal physiological processes that are happening at the time of puberty, how male and female puberty stages are undergoing all across all the pubertal transition, and then the disorders that can occur. So thank you very much for accepting our invitation. And by the way, we consider all our pediatric endocrinologists part of Pakistan Endocrine Society. SPETP and PS will continue to work in collaboration for areas of mutual interest. And actually, there's a dire need to introduce transition clinics in Pakistan so that pediatric endocrine patients are transitioned effectively to adult care. And that will actually improve their clinical care. Thank you very much.
Absolutely. And thank you very much.
Thank you, Dr. Aisha. Dr. Khadija, are you with us?
Thank you. Thank you, Sumaira, and thank you, Salman. The beauty is that you make such a complex topic seem so interesting that हर चीज समझ आ जाती है. Thank you, and thank you to Pakistan Endocrine Society for taking this up, and we are very happy that this collaboration is happening, and inshallah these webinars will be leading on to seminars and then on to further collaborations in future. Thank you, and thank you, Samara, for doing such a nice facilitation. Thank you.
Thank you, Dr. Khadija. And Dr. Sadia Salman, your comments, please.
This was a very, very wonderful and simple presentation. Dr. Salman, आपने किसी भी जेनेटिक्स की डिटेल में जाने नहीं गए, बिल्कुल इसको सिंपल रखा और कॉन्सेप्चुअल रखा और कॉन्सेप्ट बहुत अच्छा क्लियर कर दिया. क्योंकि सम टाइम पहले जब भी मैंने इस टॉपिक पे प्रेजेंटेशंस होती हैं तो जीन्स की बातें इतनी डिटेल में चली जाती है कि प्रेजेंटेशन बोरिंग हो जाती है. तो बिल्कुल टू द पॉइंट कॉन्सेप्ट पे फोकस करते हुए इतना अच्छा ये टॉपिक समझाया है, स्टार्टिंग फ्रॉम द अफ्रीका टू द एंड ऑफ प्यूबर्टी. सारा कुछ बहुत अच्छा और बहुत वंडरफुली और बहुत सिंपली बहुत क्लियर मैसेज कन्वे किया. थैंक यू डॉक्टर सलमान.
So kind of you for those nice words, and really, I keep telling people, लोग समझते हैं कि जी genetics वाले को पता नहीं क्या brain होगी और क्या समझ होगी और क्या होगा. I like to keep things simple because मैं खुद ही simple चीज समझता हूँ. ज़्यादा चीज complex हो तो मेरे भी दिमाग बंद हो जाता है. So I think we just build on knowledge slowly, and as we all learn together. So very happy to share that knowledge with you.
Exactly. Thank you. Dr. Mohsina, are you with us? Dr. Mohsina के connection का कुछ problem हो रहा है.
I am with you guys. Can you hear me?
Yes, yes, we can hear you.
Can you hear me?
Yes, we can hear you.
Sorry, I'm on the move. That's little unstable. But as usual, Salman की talk is like a wonderful. आपके लिए बहुत जबरदस्त होती है. Basic concepts से लेकर
difficult concepts that you can you can refresh your memory and you can refresh your concepts also. But what I would like to say कि हमें बार-बार इसकी revision की जरूरत होती है because such a complex topic, puberty and sexual development के जितने भी concepts हैं, they need revision again and again. So as such, it was a wonderful lecture and बहुत सी चीजें रिवाइज हुई. एक छोटा सा क्वेश्चन है सलमान से कि जब भी puberty की बात होती है या एंडोक्राइन की तो there is a concept of psycho neuro endocrinology की बात होती है especially puberty में. So what do you think is psychological चीजों की, what is the importance and what is the connection? And the other thing is जो आजकल एंडोक्राइन डिसरप्टर्स पे बहुत बात हो रही है सबसे आपके what do you think is it if days are so just very basic question I think like
I think we're losing you, Mohsina, but nahi, thank you very much. And I think I got the gist of what you're asking. So I think दो सवाल हैं. एक तो, the psychological axis and its influence, and the second is on endocrine disruptors. So, I mean, if you look at this diagram, if you look at this diagram, it's a it's oversimplified, right? And showing you कि hypothalamus पे 4-5 ही different axes हैं जो कि influence करके GnRH पे कर रही हैं. But probably there are multitudes and multitudes of neurotransmitters that go ahead. I'll just go on.
Everyone, and it was a participation that was the best part, I think.
Thank you, Mohsina. So yeah, I think यही है कि I think there are many other influences, as Mohsina pointed out, that the psychological axis probably influences it. But because they're so complex, we don't know. Okay, how will depression, how will any other, and people talk about stress, right? What is stress? And उसका जो physiologic biochemical milieu होता है stress का, वो क्या होता है? We don't truly understand it apart from the cortisol axis, which we understand well. So I think there probably are influences there, which this very oversimplified diagram doesn't do justice to. जहाँ तक endocrine disruptor chemicals का ताल्लुक है, yes, those are ubiquitous. They are present everywhere in the environment, and it's very hard to know कि वो कहाँ से आ रहे हैं. कि is it agricultural products जहाँ fertilizers और chemicals यूज़ हो रहे हैं, क्या पता मिल्क जो हार्मोनल चीज़ें दी जा रही हैं हमारी कैटल को, उसकी वजह से हो रहा है. मीट या चिकन में जो आजकल काफ़ी चीज़ें आ रही हैं कि जी उनमें हो रहा है. जो वाटर और स्ट्रीम्स जहाँ से हम पानी पी रहे हैं वो बन रहा है. फिर माइक्रो प्लास्टिक्स का सारा एक कॉन्सेप्ट आ रहा है सबके जी, जो ये फिल्टर्ड वाटर जो हम ये प्लास्टिक बॉटल्स में से निकालते हैं करते हैं, उसका जो है कुछ एंडोक्रिन इफेक्ट आता है कि नहीं आता. सो लॉट्स ऑफ थिंग्स गोइंग ऑन, लॉट्स ऑफ रिसर्च इन दैट एरिया. And again, because there are so many different chemicals and so many different influences, we don't truly understand that yet. So I think that's still work in progress. That's my answer. Thank you.
Thank you, Dr. Salman. So with this, we have reached to the end of today's program. I hope you all have enjoyed and learned something. As Dr. Aisha has mentioned, yes?
There are a few questions in the question and answer box. So maybe Dr. Salman would like to answer.
Yeah, let me check. Dr. Salman, can you view the Q&A box?
Let me see then. What I'll do is I will unshare the screen. And then I will be able to do it. Give me 1 second.
Or I can read the questions for you.
आप मुझे पढ़ दीजिए until I unshare.
So one question is, if low-calorie diet delays puberty in female, can this do the same for males?
The answer is yes. So if males are very undernourished, then they also will have a delayed pubertal axis. Although the influence is seen much more remarkably on the female. But clearly अगर एक certain level of adipose tissue और वो सारी चीज़ नहीं हुई तो वो मेल पे भी influence ज़रूर करेगा. So extreme malnutrition will clearly affect that part too.
Right. So another question is, what can we do to avoid genetic mutations?
Oh, I wish we knew. I wish we knew. I think a couple of things, right? So when you say genetic mutations, what do you mean? So if you mean recessive disorders, right, like CAH, for example, if you really want to prevent it, what you have to do is carrier screening and community education about consanguinity and cousin marriages, because that's the influence, right? These are recessive disorders. तो अगर आपको वाकई CAH की incidence कम करनी है, that's what you can do. A lot of disorders are autosomal dominant and are de novo disorders, which are basically randomly happening without us really knowing why it happens. तो I think जो recessive disorders हैं उसके अंदर तो आप prevention की चीज़ें कर सकते हैं through carrier screening and through awareness of community about consanguinity. But the spontaneous mutations are part of nature and they will continue to happen. और सवाल I guess what are the main hormones responsible for conception? I mean conception में तो it's the egg and sperm meet. तो उसके लिए तो the motility of the sperm plus the motility of the egg which is released from the ovary. तो वो जो ovarian follicle stimulation के जो सारे hormones होते हैं LH FSH वो सब ज़रूरी होते हैं. And then basically the egg travels down the fallopian tube into the sperm actually has to travel up and they meet somewhere in the fallopian tube and then come down, or they meet within the uterine cavity. So uske liye actual at the level of conception, I'm not aware of anything apart from the standard hormones. Maybe there's more research on that, but I'm not aware of those specific influences there. And I guess is gene therapy there in Pakistan currently only for spinal muscular atrophy or SMA? But पूरी दुनिया में तकरीबन I think there are only about 20 or maybe less than that, maybe 15 to 20 disorders जहाँ gene therapy बनी हुई है अभी FDA approved है. A lot more will come in the future, but not right now. What is presentation of 5-alpha reductase deficiency in the patient? So again, clinical question hai, but basically in essence, this will be an under-virilized male because आपकी जो virilization है, external genitalia है, testosterone to DHT वो नहीं होगा. So females will not have a presentation. Males will basically have either completely female genitalia अगर बिल्कुल ही 0 5-alpha reductase हुआ, लेकिन अगर थोड़ी सी एक्टिविटी हुई so they will be under virilized. So that's the spectrum and we can talk about that in future seminars when we meet. And short stature and puberty I think best to cover it under under precocious puberty because our bones jaldi fuse hongi under the influence of estrogen, तो वो उस वजह से होता है.
So ये जो pubertal disorders हैं, we will discuss this in the next program.
हाँ तो वो सारी चीजें कवर हो जाएंगी.
कवर हो जाएंगी.
And is the anatomical position of gonads also influenced by genes? With gene precisely? Yeah, I really don't know the answer to that question, to be very honest. Undescended testes HCG कम होता है उसकी वजह से हो जाती हैं और भी हो जाता है. Is there a particular gene that influences कि वो testes descend करेंगी या नहीं करेंगी? Not quite sure. How the amino acid glutamate helps in activating neurons for puberty can be used in delayed puberty patients? नहीं, there is no basically therapeutic role of glutamate in any of these things. And जो आप diet में लेंगे glutamate वो digest हो जाएगा. So this is something that is present at the neurotransmitter level. This cannot be given externally. And then are there any specific manifestations of specific gene abnormalities? Yes, there are. Obviously, and again we'll talk about that जब DSDs वगैरह की बात करेंगे तो उसमें जो है वो ये चीज़ें सामने आएंगी. I'm not sure why people are asking about palmar plantar keratosis, but I think again specific disorders की आगे बात होगी. I cannot tell you how you will get the certificate. And mutation in antitestis genes, can it be considered cases of streak ovaries with normal karyotype? Has it? I don't know a lot about this, Saima. I think this is something that we have to see. But जो antitestis genes होते हैं, they just actively suppress any testicular tissue from being made. And अभी तक सारे females में जो है वो define हुआ है कि normal physiologically ये females जब जा रहे थे वो ये genes active होते हैं. The diseases associated with these genes, I'm not currently aware of, and we can take a look, and I'll touch upon this, inshallah, when we talk about DSDs in the future. I think that's really about it, and I think we're done with all the questions.
Yes. Someone asked about the YouTube channel. PES TV के नाम से YouTube channel है हमारा. So you can subscribe for that and you can get the presentations from there. Thank you, Dr. Salman.
Chale, thank you very much, everyone. Really enjoyed doing it and looking forward to doing more.
Thank you. Thank you, everyone, for joining us today. So we will be having a physical seminar and probably at the end of the September. We will share the dates later. It will be happening in the different cities of Pakistan at the same time. And we will discuss the pubertal disorders in that seminar. So looking forward to meet you all there. Thank you. Thank you for joining.
Thank you very much. Thank you, Dr. Salman. Thank you, Sahar.
Thank you.
Thank you.
Allah Hafiz.
Allah Hafiz.